distinguished guest, Professor Wolfgang Nellen, the Emeritus Professor of Genetics of University of Kassel, Germany. Yeah, he is director for IGN TTLG 2010 till 2015 and founder of BioWiscom. I think so. Welcome again, yeah, Professor uh, Wolfgang Nellen. Yeah, maybe you many times, yeah, come to University of Jember 4 or 5 times, yeah, and you have many work, yeah, with Dr. Kartika and also Faculty of Mathematics and Natural Science. And the internal speakers, Ibu Dr. Gigi Desi Rahmawati, MKes, yeah, PhD, from Oral Implementation and Drug Development Research Groups, yeah, of Faculty of Dentistry, University of Jember, and also Ibu Dr. Renat Kartika Senjarini, from Vector Biology Research Group of Faculty of Mathematics and Natural Sciences, University of Jember. And the secretary, especially the secretary for research affairs, ya, Bapak Dr. Rondi, ya, and also the relevant center coordinator, also Bu Kartika and the team, ya, and professors, maybe professor candidates, yeah, yeah, many professor candidates, lecturer, yeah, ladies and gentlemen, whom I respect, especially from health-based faculties, yeah, the University of Jember. First of all, let us praise and thank the presence of Allah Subhanahu wa Ta'ala Almighty for the abundant grace and joy that we can still gather in this meeting without any barrier and in good health. On this occasion, allow me to welcome the fifth innovation talk series, INTAS, yeah, the fifth INTAS with the theme clusters regularly interspaced short palindromic repeats, correct? Yeah. Technology in its application for health biotechnology by our speaker from the University of Kassel, Germany, which is an established partner of the Faculty of Mathematics and Natural Sciences, led by Ibu Dr. Renat Kartika Senjarini, PhD, and the team. Yeah, it is also followed by best practice sharing by the two speakers from University of Jember. Yeah, this discussion is essential and relevant to the derivation of the vision of the University of Jember and the superiority states in the master plan for research and community service of the University of Jember, or RIPB University of Jember, ya, 2021-2025. In Indonesia, I think the use of CRISPR for testing gene therapy is not yet widespread. Yeah. Meanwhile, in many other countries, the application of CRISPR to medical therapy is still in the preclinic research stage on clinical. CRISPR technology is also received the attention of the scientific community within the last decades. Yeah. Therefore, this discussion will be exciting or add new insight for some of our researchers. Let's join the fifth innovation talk series with our guests and speakers. Welcome to follow and enjoy sharing this morning session. I hope you all are healthy and prosperous, yeah, and the lecturing will be helpful. Thank you and have a good morning. Wassalamualaikum warahmatullahi wabarakatuh.
Prof. Yuli for the speech. Dear distinguished guests, now we are going to have presentation and discussion sessions. Thank you, Prof. Yuli Witono, Pak Roni PhD, and Professor. You may join the INTAS participants. Dear ladies and gentlemen, for your information, every INTAS Innovation Talk series, there is always a sharing session. Okay, dear ladies and gentlemen, for your information, every INTAS Innovation Talk series, there is always a sharing session from the research group in University of Jember. This time, the representative from the research group in health biotechnology will do the sharing. So we already have in front of us the coordinator from Oral Inflammation and Drug Development Research Group, Dr. Desi Rahmawati, MKes PhD from Faculty of Dentistry, and the coordinator from Vector Biology Research Group, Dr. Renat Kartika Senjarini from Faculty of Mathematics and Natural Sciences. And of course, our keynote speaker, Professor Wolfgang Nelen. So the first presentation will be given by Dr. Desi Rahmawati. And before she presents her presentation, I would like to introduce her to all of you. So Dr. Desi Rahmawati lead the Oral Inflammation and Drug Development Research Group. It's a research group at the faculty level at Faculty of Dentistry, University of Jember. The researchers conduct research on inflammatory mechanism in oral diseases, how to prevent it, what are the causes, and how to treat them best. They conduct research using medical and health biotechnology. So what do they do exactly? Let's listen to the presentation. Dr. Desi Rahmawati, you have 15 minutes to present your research group work. Thank you.
Thank you. Thank you, moderator, for the opening remarks. And I would like to share. Good morning, everyone. Goedemorgen. Goedemorgen. If it's in Dutch, goedemorgen. So, good morning, everyone. Today, I would like to share with you what we are doing in our research group and what we have done and what our future perspective and related to biotechnology in medical and health sciences. So I would like to share with you what is the oral inflammation and drug development related to our research group. So presentation may start. So first of all, I would like to introduce that we are for lecture and also of course researchers and also dentists. So we are four, and I'm the coordinator. So I would like to directly to the overview of our research group. So it's mainly focusing on research on inflammatory and etiopathogenesis of the oral cavity, so how the mechanism of oral inflammation happens. And we also doing research in preventive and therapeutic diseases of the oral cavity. And we do as well experimental studies in vitro, in vivo, and also clinical trials in future. So what is actually our research group is, I would like to introduce it first, the relation between oral inflammation, biotechnology, and what we have done today. So as you might know that medical and health biotechnology is growing rapidly, and with the help of biotechnology, of course, researchers like us can better understand the underlying mechanism of oral inflammation, and then we would like to identify new drug targets and develop more effective therapies which is more biocompatible to many individuals. So I guess that this has the potential to greatly improve the quality of life for millions of people who suffer from oral inflammation. So that's actually the main aim. And what is actually oral inflammation? So as we might know that many of us, many of individual and patients suffer from gingivitis or periodontitis, which is a common condition in oral cavity. So mainly it is caused by plaque and also bacteria in oral cavity, which triggers immune response and leads to inflammation. So if left untreated, not really good, oral inflammation can progress to more serious condition and can also triggers systemic diseases like diabetes, heart disease. So there is also importance to understand the causes and symptoms of oral inflammation and take the steps prevent and treat it. And then this is what we have. Mostly of people suffer from this kind of symptoms. Uh, Gingivitis is inflammation in the gingiva, and if it's deeply untreated, then it can cause periodontitis, going into deep into the periodontal tissue. And the golden standards of these therapies is scaling and root planning to get rid of the plaque and calculus in the on the teeth, and also need a supporting therapy. So mainly nowadays still the use of metronidazole, which is antibiotics, and also for relief of pain, ibuprofen still being used many. Even though there are many kind of development like using oxyfresh, which is also developed from aloe vera, but in some of villages still use this kind of medication. So the role of biotechnology in oral inflammation related to our research group. So we use the cell culture, which is a technique used to support our research group to develop more and to investigate it more in the field of agromedicine related to oral inflammation. And we also in the future wanted to develop a new drugs and therapies for oral inflammation, which is more safety for many patients. And we also study the molecular and cellular mechanism underlying the condition. For example, gene expression analysis. In terms of genomic research, we haven't done that, but we also one of our research also using one of the tool like PCR. So current and future application of medical biotechnology in oral inflammation research, I think it's to develop new drugs therapies, as I mentioned many times in this presentation. And it's also can be used to develop diagnostic tools for oral inflammation, and also gene therapy and regenerative medicine approaches, which may offer long-term solution for repairing damaged tissue and preventing further inflammation. So what are the challenges since this is new development of therapies? Of course, also there is challenges like a concern about the safety and efficacy of new treatments, as well as issues related to access and affordability. So to address these challenges, we as researchers will prioritize transparency and accountability our work, kind of like having ethical principles and informed consent for patient and etc. if it needed. So these are our profile of research group. Our mission is contribute to improving in the quality of health of hard tissues and soft tissues of the oral cavity through promotive, preventive, and curative action by optimizing local wisdom that we have here. So our mission as well, I mentioned there, as well as our objective. So in the end, we would like to develop uh, the quality of public health through improving oral health. So this is what we have done. So we did in vitro. This is one representative research that we have done here. So related to our research group, one of example. We have done in vitro, in vivo, and also organoleptic analysis for our prototype. We developed a gel derived from Robusta coffee bean, and we already have penunjuknya mana ya Bapak ini? We oh sana. So we already have a prototype actually, and we tested wound healing and also some chemokines using immunologic assays, and also we did before doing in vitro and in vivo. Of course, we did in vitro to explore the antioxidant capacity in the polymorphonuclear blood human, and then also we also explored the bioactive component, and this is our prototype, and these are our roadmap by far. So as I mentioned many times, so our end objective is, in the end, we will have a clinical trial for the development of preventive and therapeutic oral inflammation in the field of dental agromedicine in agro-industrial communities. So these are our output by far. We published 6 Scopus articles, and we are having ongoing process for patent for coffee extract gel and also toothpaste, and these are also, we will have mouthwash, not only from Robusta coffee, but we also will develop it from green tea. And we also, in terms of community service, we have copyright 3 flip chart and 2 design of playing board for disabled children, and also we have 2 textbooks and 2 products which is already become a prototype, which is called Kofto gel, coffee robusta topical gel, and Kofta toothpaste. And these are some of our publication which is done by all of us, 4 of us, and these are one of example of our product, coffee robusta topical gel, which is indicate to reduce and relieve inflammation in oral cavity, and also to prevent allergies to dental metallic alloys that we use in oral cavity. So this is the, we also perform it last time in the exhibition when we have collaboration with US Davis. So these are Kofta toothpaste and also Kofto gel. And in terms of community service, we have done as well 2 project, which is, the first one is we educating pregnancy women to be more aware how to use antibiotics. So this is also related to our research group, which is having future perspective to replace therapies, oral therapies, with more biocompatible and safety product for everyone. So we educate pregnancy women how to use antibiotic, how to maintain oral health, and also to disabled children. So in terms of community service, as I mentioned before, that we have three copyrights right now: playing board and flip chart, and also for module how to use antibiotics for pregnancy women. And in conclusion, do I still have time?
Yes.
Okay. In conclusion, related to our research group, medical biotechnology and health biotechnology has tremendous potential to advance our understanding and treatment of oral inflammation. And also by using the power of biotechnology, we can develop more effective and personalized therapies for this common condition, oral diseases, I mean, improving the lives of millions of people worldwide. And however, we must also mindful of the challenges and ethical consideration that I mentioned before, that this is new material, so we have to be aware how the safety of these new therapies. That's all. My presentation. Hopefully, it might give you some overview about what we have done, even though it's not really genomic. But since it's also biotechnology talk, so I guess what we have done is included in biotechnology, health, and medical. Thank you, and have a good day. Good morning, everyone, and I will return it to moderator.
Thank you, Dr. Desi. Second presentation is from Vector Biology Research. So Vector Biology Research Group is a research group at study program level, the Biology Department, Faculty of Mathematics, University of Jember. They are focusing on research. They are focusing on research related to biology of vector of infection disease, with the main topic on mosquito vectors for dengue, hemorrhagic fever, and malaria. They aim to develop and produce products or technology or strategies to prevent. Thank you. They aim to develop and produce products, technology, or strategies to prevent and do intervention for these two diseases. So, Dr. Kartika Senjarini, please present the research group work in 15 minutes. Well, thank you.
Okay, thank you very much, Bu Enda. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. The Honorable Head of Research Institute of the University of Jember, and also Pak Rondi as a Secretary 3, specialized on research, and also our distinguished speakers, Professor Wolfgang Neren, who has come a long way from Germany to the east part of East Java, Jember. Can we go directly to the presentation, Mas Andika? Yeah. So let me first introduce myself. I don't have any picture here, unfortunately. Yeah, we are a research group at the study program level. I am the leader of this research group, and we have two other members who are Ibu Rika Oktarianti and Pak Subhanul Waton. So we sit at the Department of Biology. So if Ibu Desi has been giving a lot related with the human, now I'm going to bring you to the deadliest animal on the earth. Yes, and maybe you cannot imagine that mosquitoes is actually the deadliest animal on the earth. I mean, even incomparable to the lion or crocodile, or even what we know like snakes, because the numbers of people who are dying because of the mosquitoes, if you see in the slide, can I point here? Yeah, see, it's almost 7 times more than the other animals. See, yeah. Why is it like that? Even if you read one time, Times magazine has been known that mosquito is the deadliest animal on earth. Why? because if we see here, well, actually, it is not mosquito itself that is dangerous, but the thing that carried out by the mosquitoes, because the mosquitoes are a factor. That's why our research group named Factor Biology, because we learn and we do research related to the factor. A factor is anything that can transmit the disease. So there are many pathogens, many microorganisms that could be transmitted by the mosquito, if we see here. And if we see the next slide, this is an example of 4 diseases who are transmitted by the mosquitoes. Filariasis, or in Indonesia the term is kaki gajah. And then the next one is, this is the one microcephaly by Zika virus. Zika virus maybe is not so familiar in Indonesia, but if you see here how very pity the babies, because they don't have like skeleton, to cover the head. And the other 2 diseases related to mosquito, which are so like still a problem until today in Indonesia, are malaria and dengue fever. You know that Jember is also one of the endemic of the dengue fever until now. Yeah, but in the malaria cases, we have several spots in Indonesia who has this problem. And why is the mosquito is so dangerous related to these diseases? Well, the mosquito itself, actually, they don't have any intention to bite you and give you a disease, actually. But the mosquito need a blood. So the female mosquito, that's why the female one is the dangerous one. So not because the genders, but the female mosquito, they need blood from human as a nutrition supplies for the maturation of their eggs. Not only for the human, actually, also from the mammalian, other mammalians. So during this blood feeding, it is kind of facilitate the other microorganisms. So once they have taken blood from the people who has like dengue virus, or who has malaria parasites, who has Zika virus. At the same time, while the mosquito has taken the blood meal, they really need this blood. At the same time, the microorganism or the pathogen will go through this process and go to the mosquito. And when the mosquito go into the other people, then the mosquito can transmit the disease into the healthy people. So that's why in this slide, well, as I mentioned to you, although the Times already decided that the mosquito is the dangerous one, but actually it is not their intention to spread the disease. But unfortunately, some of the pathogens use this, their ability to do the blood meal and to transmit them into one person, one sick person to the other healthy person, so that the healthy person get infected and get diseases. And then what have been conducted by our research group? What can we learn from this? So our research group is actually, I'm very sorry that our roadmap is a little bit small, but we learn about the interaction between the mosquitoes as factors and the human host as I can say a victim of this interaction, and also the pathogen. The main goal, we are focusing on 2 main infectious diseases in Indonesia, which are malaria and dengue. And we wanted to do something for the prevention, as well as not only prevention, if it is possible to do intervention. If you see here our logo, so here it's very small. There is a word exploring and controlling. That is what we have been trying to do with our mosquito research. So if you see here in the previous slide that the mosquito, while doing the blood meal, they have taken the blood, and they use as mediator for this meal is the salivary glands, the saliva itself. So while they are taking the blood meal, they also transport the saliva, and the saliva apa bahasa Indonesianya? Iler ya Bapak Ibu ya. iler, jadi ilernya nyamuk. Yeah, I'm sorry Wolfgang, I use some Indonesian, it is easier. Yeah, so the saliva itself, so it can be mediated, the transmission of the pathogen. And then what can be conducted using this, this knowledge. So then there are many studies about it. So apparently the saliva help the blood meal, means it make the blood from the people is getting thinner, so not really thick, so that the usually if we have like blood feeding, it's the blood should be coagulated, right? Usually in the usual cases, but some proteins in the saliva, they help the mosquito not to get a coagulation of the blood during the blood meal. At the same time, it will make easier for the pathogen to go inside the body of the human. And the other side is the salivary components, iler component, yeah, protein especially, yeah, from the mosquito. It is help to modulate the host immune response. So the so that the human that get through this beating, they say that no, this is your friend, so please come inside to our body, everything is fine. So this is the modulation of the human immune response. So what is the idea of this basic knowledge? So the idea was, so if we can find the protein inside the saliva that mediate the transmission and enhance the transmission of the pathogen, if we could try to find that proteins and block that protein, so it could be a useful way to block the transmission of the pathogen. That was the idea of, and then coming out next term of the vaccine, usually vaccine based on the pathogen, but in this area, vaccine based of the factor components, and we call it a transmission blocking vaccine. And then this is the exploring thing on what has been done. So we did these are several research. Our very early research was published that we did make model vaccine. We inject just an extract of the salivary gland of certain Anopheles vectors into the murine model, and we use the plasmodium, and then we see that there is decreasing of the parasitemic rates, which means our animal model has becoming less sick than the one without the saliva extract. This is the very initial one, and then afterwards we try to find out what kind of proteins that are inside the saliva. So we do a lot of work related with the proteomics. So we identify the protein not only with the like SDS base or Western blot or whatever, but we did also the LCMS to find out what kind of protein inside the saliva, and then we did also how is our immune response to the protein inside the saliva or the factor, and also not only wet lab, but we are doing also, this is started like 2 years ago, to make a modeling using bioinformatics to find out how is the immunogenicity if we do it by using the artificial intelligence, can we say like that. So this is what we are doing so far. And furthermore, using this process, we find out to try to find an apirase. Apirase is one of the protein inside the saliva, which is finally we found if this protein can inhibit the coagulation, then it can maybe like another potential as a anticoagulation protein. This is also another work from our team. So from this research, from this approach, we have 1 patent and several publications, as you see in our site. And the other approach that have been conducted by our research group is controlling. So if we see here, why we have many intervention actually for the mosquito. There is like fogging and like spraying the insecticide. Why there are still many cases in Jember, especially dengue is almost every year, never stop. But apparently, it's not all the mosquito are factors. So now, why is it important to identify the factor? Because we wanted to control The bad thing is if we only identify the mosquito only using by the morphological characteristic, it is not so easy. As you see here, maybe it is not so easy to identify. Mosquito is also so complex with, they have like a sibling species, they have a complex species, like one species Anopheles vagus apparently consists of two subspecies, which is one of them is not actually a factor, but the other one is a factor. So that's why to control the mosquito and do the surveillance things, we are using the DNA barcoding approaches, and then we propose to regrouping the group of the mosquito vector, especially the Anopheles one. And then we even also like trying to design a new DNA marker for the DNA barcoding of the mosquito, as already published by our research group, the ITS primer for identification of the mosquito. So this is two approach that have been conducted by our research group, and also in the last 2 years we have also developed to do the artificial intelligence, like also bioinformatic as well, approaches to study the interaction, and also now we are also going to the microbiome of the mosquito, because inside the mosquito body apparently there are microorganisms who also have the pathogen to get survive inside the mosquito and that infected later on to the human. Yeah, I think that's all that I can present in this room. I hope it gave you kind of improvisation or idea, and furthermore a real discussion, so that we could like collaborate and improve each other. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Kartika, for the presentation. Dear ladies and gentlemen, I'm sure that some of you have something to be discussed with the two presenters, but please be patient, because we are going to have the next presentation, the keynote speaker.
Okay, okay. Stick ya, Sabet. Yes. Adanya di laptop ya. Yes, wait a minute.
Okay, while waiting for the installation for the presentation, I will introduce Professor Wolfgang Nellen first. Okay, Professor Dr. Wolfgang Nellen is Emeritus Professor of Genetics, University of Kassel, Germany. He was the director for Indonesian German Network for Teaching, Training and Research Collaboration, IGN-TTRC, 2010 until 2015. So University of Jember is already familiar for him, since our university is part of the consortium involved in this project along with other Indonesian universities. His last visit to University of Jember was in December 2022 as one of the assessors for ASEAN accreditation. Professor Nellen is also the founder of BioWiscom, a small but well-connected company dedicated to support science communication in schools and the public, but also to help scientists in teaching and research. Well, I think it will never enough to talk about his expertise, so I will stop here and invite Prof. Nellen to present about CRISPR-Cas and its application for health biotechnology. We are still waiting for the installation for the material of presentation, so well, distinguished guests, I think while waiting for Professor Nellen material, maybe you want to ask something to the two first presenter in front, please. Any of you? Any of you have question for the Dr. Desi and Dr. Kartika? Yes, please.
Yeah, thank you very much. My question for Dr. Kiki Desi.
Excuse me, sir. Could you please introduce yourself?
Yes, my name Masuri. I am from Prodi D3 Keperawatan, kampus
Okay.
Lumajang.
Okay.
My question for Dr. Desi. Metronidazole is the antibiotic or antimicrobial specific for anaerobe bacterial in case periodontitis and gingivitis. What about periodontitis and gingivitis for case pregnancy? Because in case pregnancy, antibiotic specific for here. Thank you.
Okay, thank you. Dr. Desi, would you like to answer directly?
Yes.
Yes, please.
Thank you for the question. We also investigate this kind of symptoms to pregnancy women, and it is indeed due to the hormone in pregnancy. They develop lot gingivitis due pregnancy. So what we do, it's not recommended to use antibiotics, of course, due to their pregnancy. And what we can do is doing scaling and root planning without any medication. So that's what we recommend to pregnancy women.
Recommendation symptomatic therapy.
Yes.
Okay, thank you enough.
Okay, thank you. So the presentation is ready right now. So if you have any further question, please postpone it after for the discussion session. Okay, ladies and gentlemen. Okay, Professor Wolfgang Nellen, time is yours.
I can take this microphone. You always have to use microphones. I don't know why. Okay, so no problem hearing me? No. Okay. Still no problem hearing me? All right, good morning. And thanks for inviting me here. I hope you can tell I can tell you a little bit about CRISPR, and as far as I understand, we have a lot of lecturers also in the biotechnology program. Is that right? Lecturers from the biotechnology program. So I'll try and convince you that CRISPR-Cas is something you should not be afraid of. It can be done. Is anybody working on CRISPR here? No. This is too. You are. Okay. That's good. So it's not magic. It is actually doable. It's not so expensive, and I'm absolutely convinced you should put it into your teaching programs. And not only teaching the methods, but also doing practical things. And I'll talk about that in a second. Okay, let's just see. Introduction. we are talking. We will be talking about tools. So the basic methods of CRISPR, and further tools that have been derived from CRISPR, and maybe some applications. So as I was already introduced, BioViscom is a small company. Well, yeah, it's a company for legal reasons, not to make loads of money. The CRISPR project is in part funded by the German Research Association, by the DFG, and we are collaborating with the American company Mini PCR Bio, and I'll talk about that also a little bit in a minute. What do we have? Yeah, this is what I said. Part of this seminar is funded by the DFG, where we do the public outreach project on CRISPR-Cas. Well, you probably all know these two ladies, Emmanuelle Charpentier on the left and Jennifer Doudna on the right, who got the Nobel Prize for CRISPR in 2020. I don't have to go into the details, you probably know. What they set up in 2012, the general principle of CRISPR-Cas is something like this. That's the original wheel. Where we are now is something like this. This is what you can make with a wheel. Within these last 10 years, there is a huge advance in the possibilities, in the tools, in the technologies of CRISPR-Cas compared to the onset in 2012. So development is going extremely rapid. I just included this this morning recommendation for people involved in teaching. We have a while ago developed an experiment for undergraduate students. We actually even do it at schools. It's called Knockout, and it's a very simple but very real CRISPR experiment where you can visually see what CRISPR is doing. You knock out a gene in bacteria, a specific gene. You can show that the gene is gone, the DNA is gone, and the phenotype changes, and you can show this by various ways. And it's a simple experiment. Unfortunately, I can no longer give it away for free. It's been marketed by Mini PCR Biotech, and in Indonesia, the company Plasma Unitech has the license to market this kit in Indonesia. Okay. The paper is published here, and it has all the details that was in 2019. It has all the details of the experiment. When you are clever, you can actually reconstruct the experiment. You don't have to buy the kit, but you can make it yourself. It's a question how much time you want to invest to reinvent the kit. But all the details are in this open access paper in methods. So that's up to you, but I really believe it is high time for Indonesia to start working on this, because it's important for two main fields, agriculture and medicine. So you have to get it go and get it done. Okay. Just to start out with, many of you will be familiar. I will still repeat this. CRISPR-Cas comes from bacteria. It's a bacterial immune system, and it consists of two parts. One is the CRISPR array. So this represents part of the genome in a bacterium, and the CRISPR array is a set is, well, these black symbols are repeats. They are all the same sequence, short sequences, and in between two repeats we have various sequences. So the green, the blue, the purple, the light blue, they are all different sequences, and at some point people found out that these short sequences are derived from viruses. And the bacteria who carry a sequence of a specific virus become immune against this virus. To achieve immunity, you need certain power machines, proteins, to confer this immunity. And these proteins, the workhorses of the CRISPR-Cas system, are the Cas proteins, CRISPR-associated sequences. So these are genes coding for proteins that confer the immunity. How does this work? Briefly, a phage infects a bacterial cell, and most of the bacteria will be lysed, they will die, but some may take a little bit of the bacterial DNA, insert it into the CRISPR array, and what happens then is this CRISPR array is transcribed into RNA. It's one long piece of RNA with the repeats building this little loops, and in between the different colors of viruses that have previously infected this bacterium. So this long pre-RNA is processed into small pieces, always one repeat and one specific viral sequence, and then it associates with one of these Cas proteins, building a CRISPR-Cas complex, a complex of protein and one specific RNA. So it could be the green RNA, the blue RNA, the orange or the yellow RNA. We have all these different complexes, and the CRISPR RNA is kind of the postal code for this complex to guide it, to guide the protein to an incoming virus. And when the protein gets to the virus and docks to this sequence, to this postal code, it will cut the viral DNA and destroy the virus. And this is what confers immunity. Okay, with the original detection of the CRISPR-Cas system, people jumped on this project, and within a very short period of time, many CRISPR-Cas systems were detected in various bacteria and archaea. So we now have a huge selection of different systems, CRISPR-Cas systems. They all do essentially the same, provide immunity against infecting phages, but they are set up differently. I don't want to go into the details, but here are just the three main types of CRISPR-Cas systems. I guess just to confuse you a little bit, this is a somewhat more detailed map of different CRISPR-Cas systems, but this is very incomplete. This is almost 4 years old, and there are many more systems now available. It just should impress you how many different systems have been investigated and can now be used for applications. So what really happens, a little bit in more detail, the most widely used system is Cas9, the protein. This complex is the prototype of CRISPR-Cas, and it's most widely used. So the blue outline represents the protein, the Cas9 protein, which essentially is a nuclease. It requires the RNA, the CRISPR RNA, which binds to this protein, and it requires a further RNA, which is called the tracer. Let's see. Yeah. And for practical reasons, in the lab, people have combined the CRRNA by a linker synthetically to the tracer, so we only have to deal with one RNA. And this one RNA has, parts like this, the tracer, but the most important is the green part, which is usually fits, matches to a viral RNA. But you can change this part. You can put on a different sequence, and you can target this sequence. You can use any sequence you want and target this complex to any point of any DNA of any organism that you want. And what the protein does there, it cuts the DNA. Okay, that's essentially, this is all. Properties of the protein, the Cas9 protein, it has 2 nuclease sites because it's supposed to cut a DNA, and a DNA consists of 2 DNA strands. And there are 2 distinct nuclease sites. One is called RUFC, cutting the upper strand, and one is called HNH, cutting the lower strand. Okay. The structure, what happens when you cut a DNA? Well, you should know that in your body, in your cells, DNA breaks all the time. You're sitting in the sun and you get DNA breaks. We have radiation. We have ionizing radiation all over the place, especially when you go by airplane. You get radiation, you really don't want to know how much. You get DNA breaks. What happens when the DNA breaks? Well, if nothing is done about the break, the cell will die. This is not so good, especially when you're on an airplane and you disintegrate and your body falls apart because all the cells die. No. We have repair systems that stick broken DNA together, and this is really essential. So the repair is more or less good, it works, and what happens here is when you set a DNA break at a very specific point by your CRISPR RNA, this will also be repaired. But in many cases, the repair is not perfect. You get small insertions, 1, 2, 3 nucleotides insertions, or you get small deletions just because the repair is not 100% perfect. Okay? What happens when you get an insertion or a deletion? In most cases, and when this happens within a gene, in most cases you get a frame shift mutation, which means the gene is no longer functional. In other words, when you target a specific site in a genome, you set a break, you also set a mutation. And in most cases, you will knock out a gene. There's one other thing you can do. Okay. Let's just go back. This is called, because we have ends here which are just wobbling around, and by accident they are rejoined by ligands. This is non-homologous end joining. These ends have not much in common. They are just joined more or less randomly with little insertions and or deletions. The other possibility is you help the cell to rejoin correctly. And what you can do is you provide a template, something that the cell recognizes as being homologous to the sites left and right from your cut. Okay? And then the cell will use that, do crossover, and repair in a different way. Okay? What you can also do by this method, you can smuggle other pieces of DNA in there. The cell does not recognize that there is something in between. It says, okay, this is the same. I'm on the left and on the right. I'm using this as a template, and it will incorporate the yellow part, which may be 3, 4, 5, 10 nucleotides, or an entire gene. So you can introduce new DNA into a gene. Okay, I don't want to go into the problems because otherwise it gets too long. The CRISPR-Cas complex is set up in a way that it really should cut only once in a genome. So human genome, if you do it in humans, human genome has 3.5 billion base pairs. A CRISPR RNA is specific for one specific site within these 3.5 billion. However, not everything is perfect. Sometimes the CRISPR will go to places that are very similar but not identical to what you intend. So in some cases you get so-called off-target effects. Where is it? Here. So it may cut at additional sites, and you have to be careful that this does not happen in, for example, human therapy. So what can you do with this? Just one example from agriculture, to for example, knock out a gene that keeps the roots from going too deep. When you knock out such a gene, what you get is plants with deep roots. Plants with deep roots are more resilient to dry season because they can reach water in lower levels of the soil. Just one very simple example. So just by knocking out genes, you can improve plants according to what we want, what we need for our agriculture. Yeah, we leave that out. So essentially, when you do this in plants for agriculture, what you get is a targeted mutation. So in conventional plant breeding, you're inducing mutations, you're looking for natural occurring mutations that are completely random. Yeah, mutations happen all the time, and you're looking for a mutation that by accident hit a gene that you want. This is very, very rare. You can enhance mutations by blasting the plants with radioactive irradiation. You can enhance mutations by blasting plants with chemicals. Okay, then you make more mutations and the chances to find something that you want are increasing. But you have a huge background of other mutations. Okay? But essentially, what you do with CRISPR-Cas is setting a mutation at a point where you want it. So it's targeted selective mutagenesis. So what you normally have is a cell where you get natural mutations. Natural mutations are rare, and among them there may be this red mutation that you really want. Okay? But you always have a very low background of random mutations that occur all the time. When you use chemical or radiation mutagenesis, you induce lots of mutations. These are the yellow ones. We still have the green ones which occur anyway. And among many, many mutations, there is a higher chance to find the mutation that you really want. However, you have many, many yellow mutations in the background where you have not the faintest idea what they are doing. They are just there, but you don't know where, and you do not know what they are doing. You have to select it. You have to be careful that they don't do anything bad to your plant, whatever. When you do CRISPR, we still have the green mutations that are often there. We target specifically to one locus. We get the mutation that we want, for sure. Okay? And we may have one or two off-target effects, but we can check. Okay? So this is the fastest and safest way to get a mutation that you really want. All right? Now, let's go just back once. Can you distinguish between the red mutation that you just get by chance and by waiting, the red mutation that you get by chance, mutagenesis with radiation or chemicals? Can you distinguish between the mutation, all these different mutations, if you do it by then you can get this mutation as a natural one, as a chemical, or as a CRISPR mutation. And what I did here is label one red, one yellow, and one blue. But when you see DNA, you cannot distinguish between red adenosines or yellow or blue adenosines. They are just adenosines. So you cannot distinguish, you cannot know if how a mutation was generated. And this results in a legal problem. CRISPR-Cas is no doubt gene technology. You're working on genes and you're using a technology, right? Now, in some countries, gene technology is not liked very much. And they want to put a label on. This is gene technology. With CRISPR-Cas, you can't do this. You cannot check because you cannot distinguish if the mutation was introduced by technology or if it occurred randomly. Yeah, because the nucleotides don't have a label on. I am chemical. I am made by CRISPR-Cas and I occurred naturally. They don't say, they don't tell you. You just get the sequence and you don't know how it happened. So the legal problem is mostly in Europe where people are against gene technology. There is no way to tell if a plant is CRISPR is made from gene technology or is a random natural mutation. How much more time do I have? Still a lot. Oh, great, great, great. Yeah, we still have discussion time. So what I would like to do with you now is to look at these Cas enzymes, at these proteins, and what we can learn by investigating these proteins. So one of our comic artists has an idea what such a protein looks like. In reality, it rather looks like this. So the structure is complicated, but structural biologists are excellent people. They find out exactly what a protein looks like and what it does. They learn, they can tell you about the dynamics, how proteins work. It's just really amazing. So, this is the structure, and you may remember the H and H domain and the RUF-C domain. These are the nuclease domains of the protein. They cut one and the other strand of the DNA. Here is another one, the PAM interaction domain. I may go into that in a minute. We are not going to talk about the REC domain. But what you can see is that the RNA is and the DNA are in this structure. It's difficult to see. They form base pairing between the RNA and the DNA, which means the DNA strands have to open up and the RNA invades, and only then the complex, the nuclease activity becomes activated. Before, the protein is not a nuclease. It's only a nuclease when it binds to a specific DNA. Oops. Oh yeah, this is something special. I told you that the CRISPR part of the RNA matches the a specific site in the DNA. This is not all. There is an additional site which is called the PAM domain, the protospacer adjacent motif. It's kind of a pre-selection. The Cas protein can only cut when it is targeted close to a PAM sequence. The PAM sequence is very short. It's just usually just 3 nucleotides, and it's not very strongly defined. It's something GG for Cas9. For the other Cas proteins, it may be different. So only when this matches, so the specific site, and we have an NGG next to it, only then the nuclease becomes active. Interestingly, what you of course know, base pairing is a very specific recognition. It's just RNA DNA hybridization. Here, it is not base pairing, it is amino acids specifically recognizing bases. So the PAM is recognized by amino acids, while the CRISPR RNA recognizes the sequence on the DNA. So this is just an example. What we have boxed here are the PAM sequences in some DNA, and you can now select where do I want to target, where do I want to set a cut in this DNA. Let's say this is transcription start site, beginning ATG, beginning of a gene, stop codon, end of a gene. So where do we want? We want to knock out this gene. We want to target CRISPR-Cas to a certain sequence. AGG is like NGG, that's the PAM. So we need something which is close to a PAM. There's no chance to target here because there is no NGG. We can target here, 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 or there. So we are limited to certain sequences that are close to a PAM. Only there we can target. Okay? And it's good to target early in a gene, so you disrupt, you set a frame shift early in a gene and the protein is disabled. This is a good site, and this is maybe even a better site to cut the DNA. Well, we don't need this. So, knowing the structure of the Cas is important. Knowing the structure and function of a protein is a prerequisite to modify the protein and change its function. And this has actually been done. We know where the RUFC domain is. We know where the HNH domain is. And what we can actually do is alter, change the amino acid sequence in, for example, the RUFC domain so that everything else works. the recognition, the binding of the CRISPR RNA to the complex works, the targeting to the DNA works, the binding to the DNA works, a double strand break, but it is a single strand break. So we have created a nickase, an enzyme that cuts only one strand of the DNA but not the other strand. So the DNA does not completely fall apart, but it's just dangling on one DNA strand and not on the other strand. Okay? We'll see what we can do with that later. Now you can certainly do both. You can knock out the rough C and the HNH nuclease domains, and then we have a Cas9 which can still bind to DNA at a specific site, but it will just sit there and do nothing. Is an enzyme that does nothing useful? Well, you may all know from students that students who are just sitting there and doing nothing are not extremely useful. For enzymes, this is different. And I'll show you why. Remember, this complex is still going to a specific locus in the DNA. It will sit there, not cut. It will not go away. It will just sit there stable as a rock. What does it do? Ah, no, I leave that out. What we can do, for example, in such a complex with a dead Cas9, we can attach a fluorescent marker either to the RNA or to the Cas protein and use it as a molecular probe. For example, to label a certain sequence in a chromosome. And this is what we have up here. This is a CRISPR complex sitting at the centromeres of a chromosome. It's a dead Cas9, which is targeted to centromere sequences and labeling them. So we have a new method for labeling specific sites on a chromosome. This is very convenient. What else can we do with a dead Cas9? Well, you all know the simplified structure of a gene. We have a promoter, we have a coding region, and the coding region is transcribed. Oops. Promoters bind transcription factors, and the transcription factors take care that the gene is transcribed. Now, if you target a dead Cas9 to the promoter region, this thing is sitting there and there is no more room for the transcription factors. So essentially you are blocking a gene. Okay? This is also gene technology. In this case, you do not even change the DNA sequence. You do not cut the DNA. You don't do anything bad to the DNA. You're just blocking access to the transcription factors. You have a roadblock there, and you have a functional gene knockout without doing any harm to the gene. Okay? even ways to manipulate this binding from the outside. You can induce the binding, for example, by chemicals or even by light, so that the binding only occurs upon a signal, but this is still not routine. You can do it even better, not just by blocking the access to transcription factors, by having the CRISPR complex glued to the promoter. You can even improve it. There are, as you may know, transcription inhibitor proteins. And the nice thing about the CRISPR protein, the Cas9 protein, the nice thing is you can stick all kinds of things. You can stick parts of other proteins to the Cas protein without changing its function. You just get additional functions. And when you stick an inhibitor, transcription inhibitor, like CRAP, or there is another one, let's just stay. CRAP is a well-known transcription inhibitor. When you stick this on, you will absolutely, definitely inhibit transcription. Even better than just blocking access to transcription factors. And, okay, this is called CRISPR-I for CRISPR inhibitor, transcription inhibitor. But you can also do it the other way around. You can stick a transcription activator to Cas9, activator like VP64, that's a viral protein, or the yeast protein GCN4. These proteins are much stronger activators than the usual transcription activators that bind to promoter. So you will enhance transcription of a gene by such CRISPR-A, CRISPR activation process. So you can build these proteins, or you can build these fusion proteins on the Cas9 and thus inhibit or enhance the expression of genes with a very high specificity, because you only target a specific gene. That's where the inhibitor or the enhancer will go. Should we have a break in between, or what do you think? Because people get tired, I guess.
Okay, then, ladies and gentlemen, we have coffee outside. Maybe you can grab a cup of coffee and then go back inside, and we will continue the presentation from Professor Nellen.
Yeah,
Okay, is that okay?
Is that okay?
Yes,
Yeah.
of course. You may have a cup of coffee. Grab it. It is outside this room, and then please go back again, and we will continue the
Just
presentation.
tell them how much time.
Okay, 3 minutes. No, 5 minutes is okay. Please.
7 minutes.
Yeah, 7 minutes will be suffice, okay. Ya, please. Bapak Ibu, ada kopi di luar, mungkin bisa ambil biar nggak ngantuk. Kita kembali lagi, oke? Silakan. 7 menit ya. Dear ladies and gentlemen, we are going to start the program again. If you still have your coffee outside, please come inside.
Yes, sure. Yeah, I will just give a little bit into applications. Yeah.
Thank you. So, ladies and gentlemen, will continue to listen to the presentation from Professor Nellon. So, until 11, and then we will start the discussion session. Okay? Please, Professor, time is yours.
Okay. So I'll go a little bit into applications, and these are applications in. Yeah, let's start out with some medical applications. As I said before, there are various Cas proteins, and the Cas13 is a little bit different because it targets RNA, not DNA. You may know that there are many viruses that are RNA viruses, and Cas13 is against RNA viruses. Cas13 has okay. What you can do is you can put the Cas13, the protein, or the gene for the Cas13 protein, together with a specific CRRNA, so the targeting, the postal code against the virus. You can put that in a lipid nanoparticle. That's a small, very, very, very small particle that can be vaporized and forms an aerosol. What can you do with this? It's been done in animal experiments, and this is supposed to be a hamster which is infected with a virus that goes into the lung. So this hamster is sick. You give it an inhaler and make the hamster breathe the nanoparticles, and they will go into the lung, they will go into the cells, express Cas13 and this little RNA which is targeted against the virus. And what happens? Well, we have the nanoparticle with the gene for Cas13 and the CRRNA, and this is the hamster after inhaling this aerosol. What happens is Cas13 recognizes the viral RNA and cuts it into pieces, which means the virus can no longer replicate, and there should be an improvement of the health of the hamster. And this is what really happens. I don't want to go into the details of this experiment. Again, you see there are more controls than the experimental, but what you really see is the viral load in the lung of the hamster is decreasing strongly after the application of the inhalation. Okay, so this is the viral load in the beginning. Now you give it the Cas13 and the targeting RNA, and the viral load goes down dramatically. So this is still in the experimental phase, but it looks very, very good for curing lung-borne viral infections. Okay, and this is, I mean, it's really convincing and easy to inhale something and your virus load goes down. Yeah, that's really very promising. So this is one application of CRISPR-Cas, not changing your genome, not modifying anything in your genes, but in this case, just getting rid of viral load, yeah, of a virus that infected you. Let's see what we have here. Okay, this is another application which is also extremely useful. This is Cas13a, and I have to explain what this thing does. It also has a different kind of slightly different mode of working. What it does, it also targets RNA, and when it finds a target, like here, so the Cas complex finds a target RNA and cleaves it. And after cleaving the target, the specificity is lost. And after cleaving once, the Cas13 will cleave any RNA. So by the first cleavage, it is triggered to cut everything, every RNA. What is this good for? In the cell, it will essentially kill the cell. When there's a virus, it will cut the virus once and then cut everything, and the cell, the infected cell, will just die. That's also useful. But for diagnostic, as a diagnostic tool, there's something different. So again, when a target is recognized and cleaved, then you get promiscuous cleavage of everything. So what can you do with this? You have a patient sample and you want to know if this patient is infected by the virus. Okay? Could be done in a very early stage. Very few virus RNA is in the cell. There may not be any symptoms yet, but the virus RNA is there. We mix the patient sample with an indicator RNA. That's an artificial RNA that's mixed with the sample, and this RNA has a fluorescent marker here, a fluorophore, which emits light. At the same time, on the other end, there is a quencher which quenches this light. So you have an RNA which shines bright, but it does not shine bright because there is a quencher close by. Yeah, this switches off the light. Okay, what is it? So you have an RNA that may shine, may emit light, but it doesn't. What is it good for? We have this indicator RNA in the patient sample. Let's say the patient is healthy. What happens? Nothing. The Cas13 is sitting there, does not find a target. Nothing happens. Now we have a patient that is infected. it has the viral RNA and all kinds of other RNA. Doesn't really matter. And the indicator RNA. What happens now? The viral RNA is recognized and cut. Okay? What happens after the first cut? The Cas13 becomes promiscuous and it cuts all the other RNAs, including the indicator. Now the indicator is, when it's cut, you separate the fluorescence and the quencher. They are no longer close to each other, and the light now can shine. So mixing this in and having a target results in fluorescence of the patient sample. Oops. If there is no target, if the patient is healthy, you don't get any fluorescence. If there is a single molecule of the virus, the sample will shine brightly because the indicator is cut. Got this? Understandable? Problems? No. Okay. So this assay is called the Sherlock assay and was actually developed as a diagnostic tool for SARS-CoV-2 and other viral infections. So you can detect any RNA virus. You just have to use a different CRRNA to target the Cas13 to a specific virus. Once you have the system, it's easy to adjust to any virus, may it be in humans or in animals or whatever. Easy to detect. So, I gave you some examples for tools that can be developed with CRISPR-Cas. I did not even go into the mostly applied CRISPR-Cas by changing genes, by knocking out genes, what you can do in humans, what you can do in plants, what you can do in bacteria. There's a lot of possibilities improving plants, improving or curing diseases, human diseases, by knocking out genes, by changing genes, by optimizing genes. This is just one example for plants. For example, salt resistance. We have problems with climate change. We have very dry seasons. On the other hand, we have flooding. Dry seasons, when the water evaporates, you accumulate salt, and you need plants that are more tolerant to salt, for example. And this can be done essentially by knocking out single genes. And the plant becomes more salt tolerant. And what we have here, I think it is rice, I'm not quite sure. And you can see that in the normal situation, it dies from high salt concentrations, and these at least survive. They do not yet look very healthy, but they are still alive. There is still work being done. We are doing, not we, but scientists all over the world are doing lots of work. There are plants in the pipeline that are salt resistant, that are drought resistant, and many, many other plants. A very important thing is to improve plants by making them produce micronutrients in addition to what they do anyway. And I'm not sure if you've heard about, who's heard about the golden rice? Who knows what golden rice is? Nobody knows. You don't know what golden rice is? Yeah, you do. Okay. But nobody else? Yes, okay. But all the others you should know, because it is especially important for Southeast Asia. It's a rice. Well, what is one of the nutrition problems in Indonesia? What's the most pronounced nutrition problem in Indonesia? Do you know that?
Vitamin A deficiency, malnutrition.
Vitamin deficiency, iron deficiency, zinc deficiency. And this is why 30% of the kids in Indonesia are what? Question, what is the problem with 30% of the children in Indonesia? Stunting. And stunting is a consequence of malnutrition. These kids are not hungry. They are not hungry. They get enough to eat, but they don't get enough micronutrients. Okay? So you can complement that by eating vitamin pills. Come on, go to the poor villages out somewhere and ask them, where is your next pharmacy and do you get your vitamin pills for free? No, they don't. And they get stunted kids, right? One of the solutions is to make vitamin A in rice. And this is the golden rice. It looks yellow because it has beta-carotene in. And this rice is, I think it is even approved in Indonesia. I don't know why it's not yet. It's not approved? It's not approved. Okay. This rice is essentially available for the market since 15 years. Why is it not being planted? Do you know? Why is it not being planted? If you, in part you're right, yes. You know who the what the environmental problem is? The environmental problem is NGOs. The problem is that Greenpeace spends millions and millions and millions of euros to prevent farmers from culturing golden rice. They are running campaigns. They are destroying fields in the Philippines. They intervene with the justice system in the Philippines. I don't know. I don't know, but I would suggest that this year they bribed the Supreme Court in Manila to stop golden rice, which was approved last year, and all of a sudden this year it's been retracted. the Supreme Court all of a sudden changed its mind. I may be wrong, but I believe that they bribed, corrupted the Supreme Court to implement their ideology and to keep Southeast Asian starving for nutrition. It's a scandal, but we can discuss that, and I guess I'll stop here. This is the diagnostics.
Professor,
uh,
you have Professor.
Yeah,
You
yeah.
have,
We are stopping now,
Yeah, thank you.
and this is something on our YouTube channel where you can look at videos explaining more about CRISPR-Cas tools. And now I thank you for your attention.
Thank you, Professor Nellen. Yes, thank you, Professor Nellen. Yes, of course, we have questions. Please, ladies and gentlemen, now it's discussion time. If you have any questions, please raise your hand. Yeah.
Thank you, Mr. My name is Aisa Aulia. I'm a college student under supervision of Dr. Erdia Narulita. Professionally, I will explain about our research. We are doing work in bacteriophage engineering based CRISPR-Cas9 to reduce MDR and broaden the host range. The I want to ask you some question. First, for designing a sgRNA, how do we know the optimal sgRNA design can we use? Okay. How do we know the optimal sgRNA design that can we use for CRISPR? sgRNA. For okay, okay, okay. Yeah, I have second question. In constructing an sgRNA design, uh, can we only use tracer RNA in universal, or we can use in another bacteria, or we can design construct by ourselves? the next question. Okay. uh, Based on your explanation, how to anticipate an off-target effect when we applicate in vitro method? Mm. Okay. How to anticipate an off-target effect when we apply in vitro method? Yes. 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 Okay. Yeah. Okay. Okay. Thank you, Mister.
Okay, thank you. Maybe other question from, yeah, I will give to Pak Ali Wafa first, okay?
Thank you, Mbak. Uh, Good morning, Wolfgang. I'm Ali from Faculty of Agriculture. Maybe different of most here. I'm interested about the CRISPR-Cas, and I work with that now. But I still work with Cas9 and have some problem. The first problem is about the cloning of. We use E. coli top 10, and then when we are cloning them, just small colony or not much colony, how it's we can improve that? It's my first question. Yeah. No need the plasmid. second question. Thank you for the advice. I will try in my lab. And about the CRISPR-Cas13, I never used that. It's mean we don't use any plasmid, any protocol of cloning and other, because in your example just give a inhaler to animal.
Okay. This is on patients.
Yeah.
The patient was a hamster, not a human.
Yeah.
But you do not permanently transfect the hamster. You just put in the RNA. It's like the mRNA vaccines.
Yeah.
You put the mRNA for the Cas13 in, and the mRNA is translated to the Cas13 protein.
Okay.
Okay? So for humans, you only use vectors, right? Rather you can use viral vectors.
Yeah.
But in this case, you just put in the RNA. It's a transient treatment. You don't change the genome of the hamster or the human, right?
Yeah.
Yeah, because the RNA will eventually disappear, integrate. Yeah.
So if I can make a final it's quite same with Cas9, right? The protocol.
You mean, no, with Cas9, you usually put in the complex, or you put in a vector,
Yeah.
in a vector plasmid to express DNA plasmid to express the RNA
And
and then the protein. Or you can put in the protein complex with RNA. That would also be more transient. So there are different ways. Permanently transfect
Yeah.
a cell by inserting the DNA, or
Just
transiently just putting in the complex. It works for a while and then is degraded and disappears. Yeah?
clear for me. Thank you.
Okay, thank you, Pak Ali Wafa. Other question, please? Yes.
Uh, thank you for the opportunity. I'm Rizka. Okay, I'm Rizka, research student under the supervision of Dr. Erliana Rudita. Same as Aisa, I work in bacteriophage engineering. I want to ask some question. What plasmid? What plasmid are often used and cloning techniques to insert sgRNA or cRNA until ready to use?
What plasmids are
Yes,
used
are often
I don't know of any specific thing which any specific plasmid. You can make any plasmid, essentially any plasmid useful. It depends on what organism, where do you want to put your Cas9 and your sgRNA? Into what organism, into what cell do you want to put it?
all of plasmid can we use.
You can use any plasmid, you just have to construct it, expression of the Cas9 and expression of the cRNA. But what is your target organism?
Salmonella.
You want to go into plants, into mammalian cells, or into humans, or what?
Salmonella bacteria. Bacteria. Salmonella.
Salmonella. No, so I don't know what plasmids you use for salmonella. There are specific plasmids, yes, but I don't know. Check
Yes, of course. He has to do the literature review, right? Okay. Read more, Mbak Rizka. Okay. Other question, please. Yeah, Yunita, please.
Thank you for the opportunity. My name is Yunita. I'm from Faculty of Medicine. My question is, uh, you told about the application of CRISPR-Cas in viral disease. Is there any opportunity to develop this method for non-infectious disease like disease because of genetic mutation? Thank you.
I'm not really sure. Is it already approved? Yes, of course. Okay, Professor. Let's have a short break for the prayer call. Okay, thank you. Yes. Is there any more question, ladies and gentlemen? Or is it enough? Yeah. Yeah, perhaps. Okay, I think there's no more question, Professor. Please be seated. Thank you. So I will read the conclusion for this discussion. So what the OIDD research group do is uh, the research for the health biotechnology relating to the inflammation on the oral health, right? So they recommend that during pregnancy, uh, to treat the oral cavity, we should just give the symptomatic treatment instead of antibiotics. So no antibiotic. So from Vector Biology Research Group, they develop vaccine based on the vector instead from the disease itself. And from Professor Wolfgang Nellentalk, we learned that CRISPR-Cas is doable. So we should incorporate this topic into the curriculum, and the experiments detail is available in his public in 2019, as well as the commercial kit. Well, thank you, Professor Nellen, Dr. Desi, and Dr. Kartika for the presentation. It is an interesting and, well, trigger a discussion. And I think even though some of us still maybe catching up with the CRISPR-Cas technology, but I still some of the researchers here are eager to do it in the future. Thank you, and please give a big applause for the presenters. Thank you. And dear ladies and gentlemen, before we end this program, I would like to invite the Secretary on Research Affairs, the Institute of Research and Community Services, University of Jember, Pak Muhammad Rondi, MP, PhD, to give INTAS remarks. Please come forward to join. Huh? It's okay, okay. Pak Rondi, time is yours.
Okay, thank you for MC or moderator is the same. We already in the end of this event for from the morning to the noon, and we have some remark for improving our activity, especially for today for INTAS 5 Innovation Talk Series 5. So we will have INTAS 6 in next month in the other topic. So for today's discussion, so we have maybe 4 points that we can share each other. In the first one, maybe the CSPR CA technology is the new technology for how to change the gene, but without change the sequence. This is important because the new technology will help us to develop the technology without change the sequence. Number 2, we already to research group that talking or discussing or point in this technology for Bu Desi and Bu Kartika. They also doing such research and how to improve the research using the CSPR technology. And then this technology is not only for health health topic, but also for agriculture. It also can be applied in the agriculture topic. And in the end, so Professor Wolfgang is still here up to 14 June. So maybe within 3 days, he will stay here. So if you have any more question, so please feel free to ask Professor. Maybe tomorrow he will have a workshop on maybe health faculty and also in postgraduate and also in natural science faculty, and also Professor Wolfgang is available to give some workshop related to writing in academic paper for graduate student. I hope that this event is not stop here, but we can continue in another chance, in another maybe topic as related to this topic. In on behalf of the research center, I'm say very thank you for your attendance, especially for Professor Wolfgang, also Professor Kartika, and then also for Professor Desi. We hope all of you be a professor, because professor is only the time. Yeah, I hope so. No. Going to promote. Yeah, we hope so. Okay, thank you for today, and I'm apologize if there is something a mistake from us, also from research center. So many thanks, and hope we can see you all of you in the next innovation talk series 6. So very thank you, and I will end with Assalamualaikum warahmatullahi wabarakatuh. I will return to the.
Dear ladies and gentlemen, now we have come to the end of INTAS Innovation Talk Series number 5. Again, the committee would like to express our sincere gratitude and appreciation to Professor Nelon, Professor Desi, and Professor Kartika. Amin. as well as to all of you joining this program fully from this morning. And finally, the committee would like to deliver apology for any inconvenience. We hope to meet you again in the future. Thank you very much for your attention. Have a nice day. Auf Wiedersehen, Professor. Wassalamualaikum warahmatullahi wabarakatuh. Okay. Dear participants, would you like to come forward and take a picture together?